Welcome. In this session, I'm going to discuss about the structure of a retina, and then we will also see the mechanism of vision. So here in the last session, I mentioned that the retina has three layers. So the three layers are ganglionic cell layer, cell layer, and then bipolar cell layer, and photoreceptor cell layer, right? So here, coming to this retina, so this one is the innermost layer. So this is the retina, and behind this we have is a choroid layer. So this is the choroid layer, and lastly we have is a sclera. So this is sclera, and this is the choroid layer, and this one is the retina layer. So in this retina layer we find these three layers, and how they are arranged is the photoreceptor cell layer will be close to the choroid layer. So here I'll just, so this will be the, so this region will be the photoreceptor cell layer, and below that you will find a bipolar cell layer. So this region is bipolar cell region, and below that is the ganglionic cell layer, and here is a vitreous chamber, and this is the direction of light through the lens. Okay, the light comes and falls on the retina in this way. Okay, so here, so as I told that this is the, so this layer is ganglionic cell layer. So this is ganglionic cell layer, and this one, the second one, is bipolar cell layer, and so the third one is photoreceptor cell layer. So it is in this photoreceptor cell layer we find rods and cones. So in photoreceptor cell layer, so there is a presence of rods and cones. So we have seen in the last session that rods are meant for night vision or twilight vision, and cones are meant for day vision. So that is, the cones are meant for photopic, whereas the rods are meant for scotopic type of vision. So let's see the layer here. So below that is very close to the choroid layer will be the photoreceptor layer, right? So this is the pigment layer, and embedded into this pigment layer we find the rods. So the shape of the rods is seen just like a rod. So I'll give that detailed structure of these rods and even the cones. So here the next one. So this is a cone. So that meant for detecting the red wavelength, and then so there will be cones for blue wavelength. So here I'm just mentioning the rods. So we know that in fovea region the number of cones will be more in number for having a very good resolution. Right, so this is a cone. So just I'm mentioning with the color that these cones are meant for detecting red, okay, blue and green wavelengths. So this is how you find in the photoreceptor cell layer, right? So these are the photoreceptors, right? So from these photoreceptors, so here, okay, so there will be connections with the bipolar cell layer. So these are bipolar cells, and these bipolar cells will be connecting this way, right? So here the branches from the bipolar cell layer. So this region is bipolar cell layer, and the next layer will be the ganglionic cell layer. So here is the ganglionic cell layer, and we can see here the connections. Okay, and from here we will see the nerves which all together will form into the optic nerve. So this layer is the ganglionic cell layer, and this will form the optic nerve, okay. So now let's see in detail about that rods and cones, how they will be. So here the rod will have a shape like this, okay. So I'll draw it again, right? So this is how the rod looks like, and here it has two regions. So this portion from here to here, it is an outer segment. So from here to here, it's an outer segment. So this is outer segment, and this portion from here to here, it is inner segment. Okay, and in the outer segment you find, okay, so these structures which are called lamellae, and we can see here this is the nucleus of the cell, and these will be the mitochondria, right? And this is the layer that forms okay, synapse. So this one is compared to this region which I have, okay, shown here, which will form synapse with the bipolar cell layers. Okay, then coming to the rods, I'm sorry, I mean cones. Coming to cones, so here this is the structure of cone, how it looks like. So there is a clear difference between the rod and cone, and you can see here, just like in the rods, we will also see the lamellae in the cones, and these are mitochondria, and this is the nucleus, and this will form the synapse. So just like we have in the rods the outer segment, so here this portion will be the outer segment of the cone, and so this portion is inner segment of the cone. Both the rods and cones, so both rods and cones contain light sensitive proteins. They contain light sensitive proteins, and those proteins are called photopigments, are called photopigments. So here, where are these photopigments located in these rods and cones? So here in these lamellae, so the lamellae are the locations where the photopigments are present. So these photopigments are located in membranes of lamellae. They are located in the membranes of lamellae, both in rods as well as cones. And those photopigments will be rhodopsin. So in the rods we have is rhodopsin, whereas in case of cones we have is photopsins. So rhodopsins and photopsins. So these rhodopsins are photopigments located in the membranes of lamellae of the rods, whereas photopsins are photopigments which are present in the membranes of that is lamellae membranes of cones, right? So here I will make the pigment epithelium. So this is actually pigment epithelium. So we can see here embedded into the pigment epithelium. So the now below this pigment epithelium we have is the choroid layer, and the direction of light is this way. So this is how the light falls. So the light falls actually right first on the ganglionic cell layer, then to the bipolar cell layer, and then it falls on the photoreceptors, that is cones and rods. Now here this rhodopsin is a purplish red protein, is a purplish red protein. So this rhodopsin is also called visual purple. It's called visual purple, and this rhodopsin it strongly absorbs it strongly absorbs green blue light. It strongly absorbs a green blue light, and therefore it appears in the purple color. And this rhodopsin contains a derivative of vitamin A. So it contains a derivative of vitamin A. So that derivative of vitamin A is retinol, and this retinol is we know that it, which means it is produced from vitamin A from the dietary beta carotene. So beta carotene is a precursor for the formation of vitamin A, right? And I already mentioned that this rhodopsin will help in, okay, twilight vision. So that is the scotopic vision, right? So you can take this retinol also. It's an aldehyde. It's an of vitamin A. So it's an aldehyde of vitamin A. So now here, coming to the cones. So cones, as I mentioned that, so in the lamellae they also have the photopigments. So these photopigments, as I told that it's photopsins. So the photopsins will contain retinol. So they also contain retinol and opsin. So here I'll just mention in case of rhodopsin also they have retinol and opsin, right? So this opsin is a protein, and then in case of photopsins also they have retinol and opsin. So when we see the lamellae, we know the membrane, okay, of lamellae. So we'll see here. So the membrane, and in the embedded in the membrane itself we have is opsin. So opsin is actually a membrane bound protein. So here is an opsin. So this is how we'll see. So this is the membrane of lamella, right? And here what we see is this is the opsin. So opsin is already present within the membrane, so that's why. So opsin is a membrane bound, so it's always bound with a membrane, so membrane bound protein. So right, so we'll see. So this is the opsin, and in contact with the opsin present is retinol. So here, so this green color portion is retinol. So when light falls, what happens is the retinol will separate from opsin, and this results in the generation of action potentials, right? So when retinol dissociates from the opsin, so when the light falls, the retinol will separate from the opsin, and because of this there will be structural changes in the opsin, and that results in the generation of action potentials, and those action potentials will travel okay, through the photoreceptor cell, and finally they come to the synapse. And from the synapse, those action potentials will travel to the bipolar, and from the bipolar they travel to ganglionic cell layer, and from the ganglionic cell layer, so the nerves will carry through the optic nerve to the brain. So this is how the vision in human beings takes place, right? So here, those impulses which are generated in the retina are actually carried to the visual cortex of the brain. So impulses are carried to visual cortex of brain, so where they will be analyzed, and actually the image that falls on the retina will be inverted, but the image will be made upright in the visual cortex region of the brain, right? And that is the reason why we will be able to see the object as it is, right? So here I'm showing you a very much clear picture. So you can see here, this is the pigmented layer. Okay, so that's a pigmented epithelium, pigmented layer. Okay, and below that you will find the rods. So I mean, so here are the, okay, so it's a mistake here, so it's not a rod, and here these are, okay, rods, and these are cones. So these are cones and these are, okay, rods, and below that you see the bipolar cell layer. So these are all the bipolar, okay, uh, cells. So bipolar cells here, and these bipolar cells are in connection with the ganglionic cells. So this is the ganglionic cell layer. Here, one ganglionic cell layer will have many connections with the bipolar cell, and one bipolar cell will have many connections with the, uh, right, the rods or cones. So that is about, that's all about the eye. Then next we'll see about ear. Right. So before I go into ear, I would like to tell a few more points which I thought to tell you. So one is the muscles involved in movement of eye. Muscles involved in movement of eye. There are total, right, three pairs. So three antagonistic pairs means which work in opposite. So three antagonistic pairs of muscles are present connected to the eye. So there will be one lateral rectus and medial rectus. So these two will work antagonistic. Okay, so these two are muscles. Okay, the next one, superior rectus, and another one called inferior rectus muscles, and then third one is superior oblique, superior oblique, and another one called inferior oblique muscles are located. So let's see this in a picture, right? So you can see here the lateral rectus. So this is the lateral rectus, and opposite to that is the medial rectus. Okay, above you have is a superior rectus. So this is the superior rectus, and this is inferior rectus, which will help in moving your eye, okay, up and down, whereas the lateral and medial will help the movement of your eye horizontally. Okay, whereas the superior oblique, so you can see this is the superior oblique, and here is inferior oblique. So these will help in movement of eye in a diagonal. So these are total, okay, six muscles which are connected to eye. So total six muscles connected to the eye and help in the movement of eye. Right, so that's it about the eye. Next we'll see about ear. So next we'll see about ear. So ear has actually two functions. One, hearing. So that is auditory in function, helps to receive sound waves, and it also has another function, that is maintenance of body balance. So maintenance of body balance. So it is also the function of ear. The next, let's see here, ear is divided into three parts. So that is external ear, then middle ear, and then inner ear. So the external ear will be consisting of different parts like pinna, and then external auditory meters, external auditory meters. Okay, and coming to the middle ear, we have three smallest bones called the malleus, then incus, and then stapes. And coming to the inner ear, it has labyrinth. Then here one more I'll include under the external ear, the tympanic membrane. So this tympanic membrane is also called eardrum. So this portion also I will include in the external ear. So you can see here the external ear, okay, has a pinna. So this is the pinna, and that leads into an external auditory meatus. So this is, okay, so external auditory meatus. And at the end of this is the presence of the tympanic membrane. So that is the tympanic membrane. Okay, and behind the tympanic membrane we will have those smallest, okay, bones. So located, so these are the smallest bones in the body. Okay, uh, then later we will see the inner ear, so that will be the right labyrinth. So later we'll see in detail about the labyrinth. Okay, that will be containing vestibule and cochlea. So this coiled portion here is cochlea. Okay, and this is the middle ear, so consisting of this one as a malleus, and this is incus, and this is the stapes that is connected to, okay, the inner ear. So here the external auditory meatus contains very fine hairs. So it consists of fine hairs inside, and it also has wax secreting glands. So the external auditory meters, it has fine hairs, plus it also has, okay, wax secreting glands. So these wax secreting glands are called ceruminous glands. So they are called ceruminous glands. So they are also present, okay, in the external auditory meters, which help in preventing the dust and foreign particles entering the ear. And coming to this pinna, so this pinna is also called auricle. So it's called auricle, right? And this is made up of elastic cartilage. Is made up of elastic cartilage, and that is covered by skin. Elastic cartilage covered by skin, is covered by skin, right? And coming to this tympanic membrane, so here the tympanic membrane, uh, it is composed of connective tissues. It's composed of connective tissues, okay, that will be covered by a mucous membrane. So you'll see here, so how the tympanic membrane, right? So this is a mucous membrane, and so inside is connective tissue, and then the whole is covered by skin. It's covered by skin. So this is how you will see. So the outer one is skin, and below that is the mucous membrane, and the innermost will be the connective tissue, is connective tissue. So this is how the eardrum, right? So the parts of the eardrum. So here I show you the picture in real. So here this is the pinna, that's called auricle, which I have said. So this is auricle, right? And is made up of elastic cartilage and is covered by skin. And you can see here, this portion is the external auditory meters that ends with the tympanic membrane, and behind the tympanic membrane are three smallest bones. So this first one will be the malleus, okay, and the second one will be the incus, and the third one is stapes, that is connected to inner ear, okay. And arising from that inner portion of the ear, we have the nerves. So later we'll see about those nerves in detail, and all these structures are embedded in bone. So you can see here, this is all covered all around by bone. Now coming to middle ear, so this middle ear is also called tympanic cavity, also called tympanic cavity. And this is the middle ear will be a small air filled cavity which will be present within the temporal bone. So this is a temporal bone, okay. So it's present in temporal bone, located in temporal bone, and this is an air filled cavity. So it's an air filled cavity. So you can see in this picture, so this portion is, okay, so this portion is the middle ear, and this middle ear is connected, okay, with the buccal cavity through a tube called eustachian tube. So this eustachian tube actually helps in, okay, providing the, okay, enough pressure, so that will be equal with the external pressure. So the pressure, okay, outside this tympanic membrane and, okay, behind the tympanic membrane, okay, should be almost same, so that there is a free movement of the tympanic membrane. So otherwise any blockage to this eustachian tube will not make the tympanic membrane to move freely, and the person will not be able to hear the sounds properly, right? Right, so this eustachian tube connects middle ear, so it connects middle ear with the buccal cavity, middle ear with the buccal cavity. Okay, and I told that this middle ear, it consists of three bones. It's called the malleus. So malleus is called hammer bone because of its shape like a hammer, so it's called hammer bone. The second one is called incus, and this incus is called anvil bone because of its shape to anvil. And then stapes is called stirrup, and its shape to that of stirrup, so they have named it, okay, stirrup. So these are three smallest bones. Okay, and I told you that the first one is malleus. So malleus is attached behind the tympanic membrane, so this is the malleus. Okay, and the malleus is attached to the incus, so this one is incus, and incus is attached to the smallest bone that is called stapes, and stapes finally attaches to, okay, a part of the inner ear, so that's called it's connected to oval window. The stapes is connected to oval window, so it is connected to oval window. This oval window is called fenestra ovalis. It's called fenestra ovalis. Okay, so that will be the portion of the right inner ear. And I already told you that the pressure on the tympanic membrane should be almost same on either side, so that will be helping by the eustachian tube, so that helps in equalizing the pressures on either side of the tympanum. Right, so this is small information on middle ear. So now let's look at the inner ear. So coming to inner ear, so this inner ear, so in the inner ear, so there is a fluid filled portion. So the fluid filled portion of the inner ear uh, is called labyrinth. It's called labyrinth, and this labyrinth has two parts. Okay, so one, it is called bony labyrinth, and then another one called the membranous labyrinth. Membranous labyrinth. Okay, so there is a fluid filled portion of inner ear, so that's we call labyrinth, and it has two parts, bony labyrinth and membranous labyrinth. Now the bony labyrinth is actually a series of channels, is a series of channels. Okay, and is divided into three sections, and is, okay, divided into three sections. So those three sections are cochlea, and then vestibule, and one more part called semicircular canals. Okay, so out of these three parts, it is the cochlea that actually helps in the actual one that helps in the mechanism of hearing. So this is the one that is responsible for, okay, hearing. So whereas the other two, the vestibule and semicircular canals, will help in balancing our body, and even knowing whether we are in motion or not. So these two will be concerned in that process. So later I'll discuss in detail so what will be the function of these vestibule and what will be the function of these semicircular canals in detail, including with the cochlea. So first we will see about cochlea. So this cochlea is the coiled portion of labyrinth. And when you see, so this is how it will be. So here this is the base of the cochlea, and this will be the apex of cochlea. Okay, and inside the cochlea, so if we take a transverse section of cochlea, okay, so the cochlea will show three tubes running okay, parallel to each other. So there are inside cochlea, so there are three tubes. So those three tubes will be one called scala vestibuli, so another one is called scala media, and another one called scala tympani. So here I'm showing you the transverse section of cochlea. So you can see this portion is the apex of cochlea, okay, and this will be the base of cochlea. And if you take a transverse section, so at this point, so this is how it looks like, right? So we I told that this membranous labyrinth is embedded in the bony labyrinth. So here you can see the bone are covered, and inside it's lined by the membranes, and we find there are three canals. So there are three canals passing through the cochlea. So one, it is called scala vestibuli. So you can see this portion is scala vestibuli, right? And below that is scala tympani. So it's called scala tympani. And here there is a, right in between scala vestibuli and scala tympani, we have is a cochlear duct. So there is a cochlear duct. Of course, this cochlear duct is called scala media. It's called scala media, right? Right, so scala media, scala vestibuli, and scala tympani. So this scala media is also called the cochlear duct. So this scala vestibuli, so this scala vestibuli, we can also call it a vestibular duct. So vestibular duct, whereas scala tympani, we can also call it a tympanic duct. It's called tympanic duct, right? So here the scala vestibuli and the scala media, so these two are separated by a membrane, so that's called a Rezners membrane. So it's separated by a Rezners membrane, whereas the scala media and scala tympani, they are separated by the basilar membrane. So here you can see the basilar membrane, right, located between the scala media and scala tympani, and between the scala vestibuli and scala media we have is Rezners membrane. So here the scala vestibuli and scala tympani, so both of these are filled with a fluid called perilymph. So the scala vestibuli and scala tympani are filled with a fluid called perilymph. filled with a fluid called perilymph, whereas only the scala media, so this is filled with a fluid called endolymph, is filled with the endolymph, right? So this is perilymph, and here also it is perilymph, and whereas the scala media will be filled with the endolymph. Now at the base of cochlea, at the base of cochlea, the right, the scala tympani. So at the base of the cochlea here, okay, the scala tympani, I mean the scala tympani ends with the round window. So there is a round window, it ends with a round window, while the scala vestibuli, so this scala vestibuli, so this one will end with the right oval window. Those things we will see in detail in the next session.